Do you ever wonder why some neurons can survive for decades, while others slowly degenerate over time? Peripheral neurons are some of the longest and most fragile cells in the human body. To stay alive, they constantly rely on efficient intracellular transport and stable cytoskeleton organization. So, what happens when that intracellular transport system begins to collapse? My name is Yue Yu, and today I'm excited to share our work published in Embryo Molecular Medicines from the lab of my mentor, Dr. Zhan Zhiqiang. Our study investigates how mutations in the INSC gene may destabilize neuronal organizations and contribute to Charcot-Marie-Tooth disease type 2, or CMT2. And thankfully, instead of experiments on humans, we let Bruce Fries suffer for science. Alright, let's kick things off with a disease that's called CMT2. Think of your peripheral nervous system as a high-speed electrical highway running down your leg. In CMT2, that highway starts crumbling at the far end. The axons, those long nerve fibers, begin to fall apart. Patients might notice their feet feeling numb, their walk getting a bit wobbly, and muscles slowly losing strength. And here's the real mystery. Over 70% of CMT2 patients still don't have a non-genetic cause. So yes, it's like trying to solve a crime when most of the suspects are still unknown. So here's where the plot thickens. We have met CMT2, the nerve damage valence, but now we found a new suspect hiding in the genetic lineup. Meet the INSC genes. It's been quietly minding its own business until we discovered a sneaky mutation called M70R. This mutation showed up in the family with a slow, progressive nerve damage, wobbling walking, and that classic distal sensory loss. It's like finding the missing piece of a puzzle that's been frustrating neurologists for years, except this piece was hiding under the couch for the whole time. Now, this is where things get really interesting. The INSC gene turned out to be living a double life. For years, scientists thought INSC was just a developmental gene, helping embryonic stem cell divides neatly like a well-organized kindergarten teacher. But surprise! It doesn't retire after birth. INSC keeps working hard in adults, maintaining the nerve internal scaffolding, those microtubules that keep everything running smoothly. So it's not just a baby gene anymore. It's more like the one employee who never take a vacation, quietly keeping the nervous system in shape long after the brain first contraction phase. Alright, picture this. Inside your neurons, there's a construction crew hard at work. We've got pottery and LGN, the anchors and the motor recruiter, making sure everything in place. Then there's INSC, the foreman, the one shouting, "Hey, let's keep this highway stable, people." Together, they form the PIL complex, which acts like a scaffolding to keep the microtubule highway solid and functional. Without them, the nerve internal structure would be like a construction site with no foreman. Chaos everywhere. So basically, there's the unsung heroes of nerve maintenance, tiny but mighty. Now, meet our star patients, the fruit flies. Tiny, adorable, and surprisingly helpful. These little guys have bigger neurons, basically track sensors in their leg and walk a lot like ours. Perfect for studying how we sense the movements and balance. To mimic the human M70R mutations, we gave them the flies a matching genetic twist, K305R. So yes, we've turned the fruit flies into the miniature model of human neuropathy. They may be small, but they're doing big science. Alright, time to put our tiny applets to the test. Here's how the climbing assays work. We give the flies a gentle tap to send them to the bottoms of the vial. And don't worry, it's more of a wake-up call than the punishment. Then we start the clock, 10 seconds, and watch their natural instinct kickings. The flies hate being low, so they climb upwards against gravity, showing off their motor skill. Finally, we measure how many reach the 8 cm mark. The higher they climb, the better their motor function. It's basically the fly version of a fitness test, and no gym membership required. So what happens when we put our mutant fly through the climbing test? At first, they were doing great, spriting up the vial like Olympians. But as the week went by, things start to change. By week 3, the climbing success dropped dramatically. The healthy flies keeping crossings, while the mutants, they just say they lost their grip, literally. This tells us that the mutated INSC protein doesn't cause immediate trouble, but rather an adult-onset decline in motor control. Next up, we are taking fly movement analysis to the next level, literally brain by brain. We filmed our flies walking in the shallow chambers at 500 frames per second. That's faster than most slow-motion sports replays. The software then tracks each leg to see exactly how they step. If their gaits get wobbly or uneven, we can spot this instantly. And now we're getting fancy. We use CRISPR to create a fly model that perfectly mimics the human M70R mutation. At week 1, everything looks normal. They work fine, footprints are neat and tidy. But week 2, things start to go downhill. Their step gets irregular, like they're losing rhythm. And by week 3, it's chaos, uncoordinated leg movement, massive footprints, and serious gait problems. So why do our Newton flies lose their balance? Turns out, it's not just bad coordination. Their fecal neurons, the fly version of muscle spindle, are literally dying. We track this using fluorescence marker, DAPI for helping DNA, and PI standing for damaged cell. At first, everything looked fine, bright and healthy. Then, as INSC starts falling, PI sneak in, signaling trouble. Finally, the cell goes full meltdown, membranes rupture, and the neurons are just gone. It's a tragic but fantastic story. As the flies age, their mutant INSC collapse triggers necrotic neurodegeneration that perfectly match their declined climbing ability. Now, let's zoom inside the neuron and see what's really going wrong. In healthy cells, tubulins forms beautiful, straight microtubule highway, smooth track for transporting everything the cell needed. But when INSC fails, those highways start to collapsing. Tubulin clumps together, forming messy tangles, like a traffic jam with no access. Now, it's time to connect the dots, or in this case, the puzzle pieces. In healthy neurons, PAR3, INSC, and LGN fit together perfectly, forming the PIO complex that clamps onto the microtubule and keeps it stable. But when the M70R mutation hits, it physically alters the INSC proteins, and each one puzzle pieces suddenly change shape. The clamps break, the complex falls apart, and the microtubule tracks start to crumble. So we have seen the cellular highway collapse. Now the big question is, can we fix them? Enter Tuxo, our microtubule stabilizing hero. Think of it as a molecular duct tape. If the tracks are falling apart, maybe we can just patch them up. Finally, some good news. After all the cellular chaos, we found a way to rescue the highway. By treating the mutant flies with Tuxo or a similar microtubule stabilizing drug, we could actually rebuild the broken track. So here's the big picture. We have pieced together how INSC isn't just a developmental gene. It's a lifelong guardian of the nerve health. In adults, it teams up with the PAR3 and LGN to maintain the peripheral nervous system scaffolding, keeping those microtubule highways strong and steady. When this system fails, we get CMT2, where axon degenerates and motor control collapse. But the exciting part is we can rescue this by stabilizing the microtubule and restore the function. And here's where our journey ends, but the host begins. We now know that INSC is vital for the adult peripheral nerve health. By understanding how this mutation breaks the cell's scaffolding, we've opened the door to target therapeutics. It's a beautiful reminder that even the tiniest molecule discovery can lead to the enormous breakthrough. So next time you see a fruit fly buzzing around, remember, it might just be helping us to cure neuropathy. Thank you so much for your listening.